Different cell type function as antigen presenting cells to activate naive T cells or previously differentiated effector T cells. By naive, we mean any cell which is never exposed to an antigen, so it's new to it. And by differentiated means who has already earlier been exposed to an antigen. So the antigen presenting cells can be any nucleated cells in our body, but dendritic cell, macrophages and B cells are called professional antigen presenting cells or professional APC. Dendritic cells take up antigen and present the peptide to naive T cells, which results in its activation, that is clonal expansion and differentiation into effector T cells. So these dendritic cells have MHC2 molecules on their surface and thus they stimulate T helper cells. Similarly, macrophages can also intake antigen and present it to effector T cells. In this case, they are not naive, that is, they result in their factor functioning. In this case, macrophages are displaying MHC class II molecule on the surface and they are activating effector T helper cells. This T helper cell then release some cytokines which results in causing these macrophages to digest the engulfed microbes. So it results in cell mediated immunity by activation of macrophages. B cells can also recognize those antigen and these then have MHC class II molecule on the surface which then activate T helper cells or effector T cells which activate B cells and results in antibodies production or humoral immunity. So these antigen processing and presentation via antigen presenting cells may result in naive T cell activation, cell mediated immune response as well as antibody mediated or humoral immunity. Epithelial and tissue resident dendritic cell capture protein antigen and transport the antigen to draining lymph nodes. These dendritic cells can ingest infected cell and present antigen from these cells to CD8 T cells. That is a process called cross presentation. So these dendritic cells have MHC class II molecule in the surface and their expression is constitutive. That is, it increases with maturation as well as with amount of interferon gamma. The expression of the co-stimulator is also constitutive, that is, it increases with maturation and it is inducible by interferon gamma. And the principal function of dendritic cell is initiation of T cell response to protein antigen, that is, priming. Macrophages also display MHC2 class molecule, but they have less, the expression is low or negative. However, it is inducible by interferon gamma. Similarly, the co-stimulators are inducible by LPS, that is the PAMs, the interferon gamma, and interaction of the co-stimulator on the surface of T cells and the APC. And the primary function of macrophages is to initiate cell-mediated immune response, that is T cell enhanced killing of phagocytotic microbes. B cell lymphocytes also have MSC class II molecule on the surface and their expression is constitutive and it is increased by interleukin 4. The co-stimulator expression is induced by T cells and antigen receptor cross-linking. So the primary function of B lymphocytes as antigen presenting cell is to induce humoral immune response, that is activation of helper T cells which then induce B cell activation and antibody formation. Vesicular endothelial cells in humans express class II molecules which are inducible by interferon gamma. and their expression is constitutive. And the primary function is to present antigen to blood T cells that become adherent to the vessel wall. That is, it helps in promotion of activation of antigen specific T cell at the site of antigen exposure. Processing of protein antigen involve conversion of antigen into cytosol or internalization from the extracellular environment and loading onto MHC molecule and their presentation on the cell surface. So the MSC1 molecule or two molecules are basically bound to the peptide before they are displayed on the surface. So as we have already studied that MHC1 molecule display antigens which originate in the cytosol, and MHC1 molecule present these antigen the peptide to CD8 T cells or cytotoxic T cells. On the other hand, uh, the peptides which are extracellular in origin are displayed on the surface of or the cleft of MHC2 molecule and are presented to CD4 T cells or T helper cells. Antigen processing and presentation can take place by three major pathways, that is MHC class 1 pathway, also called endogenous pathway, or MHC class 2 pathway, also called exogenous pathway, and by cross presentation. So in the first pathway, MHC class 1 pathway, as you can see, the antigen is endogenous in nature, that is, it originates from the cytosol. This antigen is then degraded into peptides. These peptides are loaded onto the surface of MHC1 class molecule, and then this loaded molecule is then displayed onto the cell surface of antigen presenting cells. So in class 2 pathway, the antigen is generally exogenous in nature and it is ingested by the cell via endocytosis, and then it is degraded to produce peptides. These peptides are then loaded onto MHC class 2 molecule, and then this loaded MHC class 2 molecule is displayed on the surface of the antigen presenting cell. In some cases, what happens, the antigen is exogenous in nature, but it is presented by class 1 molecule. This is called cross presentation. In this case, the exogenous antigen is taken up by the cell by endocytosis, but it escapes the endosomes and ends up in the cytosols. Now it is taken up by the MSC class 1 molecule and are displayed onto the surface of APC. So, first pathway, that is the MSC class 1 pathway and class 2 pathway, can be taken up by all dendritic cell. However, cross presentation can only be taken up by the CD8 dendritic cells. Now let us discuss each of this pathway in detail. So first is endogenous pathway of antigen processing or MHC class 1 pathway. So the source of these proteins which are to be presented by these pathway are cytosolic in origin. So that is, they synthesize within the cell, and in some cases they may be phagocytosed and are transported into the cytosol. And there are some bacteria which have the ability to escape the vesicles and move into the cytosol and result in cross presentation, as in case of Listeria monocytogenes. So antigen from cross presentation are also displayed by MHC class 1 apart from the one which are synthesized within the cell. Apart from the peptides which are foreign in origin, the MHC class 1 molecule also displays self protein which are misfolded, and they are translocated into the cytosols and are degraded by a process called endoplasmic reticulum associated degradation. The first step in endogenous pathway processing is the proteolytic digestion of cytosolic proteins. So proteolysis by the proteasome is the major mechanism for generation of peptides from cytosolic protein antigen. The proteasome performs a basic housekeeping function in cell by degrading and any damaged or improperly folded proteins. On average, 6 to 8 amino acids are being added into the elongated chain of amino acid every second, and out of these, approximately 20% of the newly synthesized proteins are misfolded and need to be eliminated. Similarly, defected ribosomal products and affected proteins are also ubiquitinated and threaded through proteasome. However, there are some proteins which do not need proteasomes or ubiquitination to be presented by MHC1 molecule. They can be degraded by the cytosolic proteases. For example, signal sequences of membrane and secretory proteins are cleaved by signal peptides. The second step of the endogenous pathway involves the transport of the peptide generated in the cytosol to the endoplasmic reticulum by a specialized transporter which is called transporter associated with antigen processing or TAP. It helps in transport of low molecular weight proteins and it is ATP dependent. So the newly synthesized MHC1 molecules are present and that are available to bind to these peptides. These TAP protein associate with tepecin, which also has affinity to newly synthesized empty MHC1 molecule awaiting the arrival of peptides. The final step in endogenous pathway processing is the assembly of peptide and MHC1 molecule complex and its display on the surface. So the peptide which is translocated into the endoplasmic reticulum binds to the MHC class 1 molecule and which is associated with DAP as well as tepacin. This MSC class 1 captures peptides rapidly and becomes stable and then it is ready to be displayed on the surface of the antigen presenting cell. So on the surface, the CD8 co-receptors bind to the non-polymorphic region of the MSC class 1 molecule and trigger activation of T cell. There are several viruses which have evolved mechanism to bypass MHC1 assembly and peptide loading, and thus they evade the immune system. This figure summarizes the whole process of class 1 MHC pathway or endogenous pathway of antigen presentation. As you can see, the antigen which is cytoplasmic its origin, it is then degraded by means of proteasomes which chops it down into peptides. Similarly, if an antigen which has originated from outside but has been intaken by the cell by means of phagosomes, and if it escapes the phagosomes, it becomes cytosolic, and now they can be taken up by the proteasomes and can be degraded into peptides. These peptides then move towards the endoplasmic reticulum by means of TAP molecules. These TAP molecules, along with tepecin, binds to the MHC class 1 molecule. These class 1 molecules then uh, is loaded up by the peptides, which are then molecules. These molecules are then um, released outside by means of exocytotic vesicles, and they are displayed on the surface. On the surface of the antigen presenting cell, they interact with the cytotoxic C cells or CD8 T cells, as the CD8 receptors bind to the non-polymorphic region of the MHC class 1 molecule.